Hello everyone, welcome to the ITLS Academy. Today we are going to perform another experiment, that is your direct staining. Okay, so before starting our experiment, we have to know that what is this staining. Okay, so simply staining is what? Staining is defined as a it comes under the category of microscopic technique which we use to enhance the contrast in microscope image. Okay, simply what we have to do, we use a staining. Um, in staining, we use a stain, different stain, and these stain increase the contrast in microscope. Okay, which means that it increase that image that's shown inside the microscope. Okay, we all know that this microorganisms are what? Microorganisms are colorless, which means that we cannot see it without any stain. Okay, if something is colorless, then how we see it without any stain? That's why this staining are used, which give a color to the microorganisms and also highlight that cells. Okay, when these what is the role of this staining? These staining highlight that cell, then that cells are visualized when we see under the microscope. So this is a concept of the this is a concept of the staining. Now the today the experiment that we are going to perform is a direct staining. Direct staining are also known as a positive staining. Why? I'm going to explain you. See, this direct staining have a what? They have a positive charge portion of a dye. Okay, when we use a when we use a dye in a positive staining, then that dye have what? They are a positive charge, and mostly the bacteria cell are what? They are a negatively charged. Okay, so when they both are come in contact with each other, they will attract towards each other. Okay, so these dye color the bacterial cell. Okay, the dye which we use in a direct staining, what they do, they colored the bacterial cell, because in direct staining, the stain that we use, they are what? They are positively charged, and the cell or bacterial cell are negatively charged, and both are attract towards each other. That's why it give a color. Okay, so here in direct staining, we use a basic dye. Okay, for example, we use a methylene blue, saffronin, crystal violet, malachite green. They are what? They all have a positively charged portion. Okay, that's why when they come in contact with the bacterial cell, they highlight that bacterial cell. Okay, so you can use any one of them dye, that is your methylene dye. Either use saffronin, crystal violet, and malachite green. Here, the dye that we are going to use is a methylene blue stain. Okay, this is your methylene blue stain. Now, what is the procedure? Simply what we have to do, see, what we have already a culture, that is we perform it with the help of the pouring method. How we do this pouring, or how we prepare a media for this, and how we do a serial dilution, the videos related to this are already uploaded in our account. You can visit our YouTube channel and see the video related to this. Okay, then you can better understand. Now already we have a culture, that is pour plate method, and we have 10 to the power minus 4 serial dilution. Now what we have to do, we take a uh, that slide, and with the help of the inoculating loop, first what we have to do, we sterilize that inoculating loop. How we sterilize it? By passing it through the heat. Okay, then we cool down it, then after that we take a smear with the help of that inoculating loop, and then we transfer that smear culture into the slide. Okay, then after that we pass that slide from the flame. Then for a fixing purpose. Then after that we cool down that slide. Then after that we add our stain. Okay, here what we are going to add, we are going to add a methylene blue. Then after that after 5 minutes we wash that stain with the help of the distilled water. Then we dry it. Then after that we examine it under the microscope. Okay. Okay. Now when we stain it with the methylene blue and we see under the microscope, it appear a blue in color. Now let's start our experiment. First of all, what we have to do, we have to heat it. Okay. Now first of all, heat the burner. Now first of all, we have to heat fix the inoculation loop. Okay, so that it will be sterilized. Then cool down it, and at that time we have to take out the slide. Okay, this is our last slide. Now with the help of the inoculation loop, we have to take out the smear like this. Take your inoculation loop and take a smear. Okay, don't take a agar. Now put it into the middle of the slide. Then you have to pass it to this slide through the flame for a fixing. Like this. Now you have to cool down it. After that we cover that smear with the help of the methylene blue for a 5 minutes. Then after 5 minutes we wash it with the help of the distilled water, and then we examine under a microscope. Okay. Now add a smear. Now wait for a 5 minutes. After 5 minutes, what we have to do, we have to wash this methylene blue with the help of the distilled water. Heat fix is very important because it will fix the smear with the slide. Otherwise, it will float with the distilled water. Now you have to air dry it, and after air dry it, we will examine it under the microscope. Okay. Now at the end of the video, we will attach the image of the image directly taken from the microscope for your better understanding. Then you can see that how the cell are visualized under the microscope. So this is all about the positive staining or a direct staining. I hope you will understand. But if you have any query related to this experiment, you are free to ask me in a comment box, or for a further detail, you can follow the ITLS Academy on different platforms. That is your Instagram, Facebook, Twitter, YouTube, WhatsApp, and LinkedIn. And subscribe our channel ITLS Academy on YouTube and get our app from a Google Play. You can also visit our website, that is www.itlsacademy.com. Thank you for watching. Please like, share, and subscribe our channel.